My name is Gabriel Bronk, and I do computer simulations of chromosomes. I work with Professor Kondev, and we collaborate with Professor James Haber, whose lab conducts experimental studies of chromosomes in yeast cells. Our findings could inspire new microscopic materials that can change their shape when given an input. The chromosomes inside yeast cells are like tiny ropes, and due to their thermal energy, they're constantly wiggling around, forming different shapes. Yet despite this randomness, we've determined that yeast cells do have some control over the chromosome shape. They can change the shape when necessary. How do they do that? I'll tell you. Enzymes can attach phosphate molecules to proteins at a particular spot on a yeast chromosome. I'll represent the phosphate molecules with this piece of Velcro. This other piece of Velcro represents a protein that binds to a different spot on the chromosome. And this protein also binds to the phosphates. Thus, the two parts of the chromosome get stuck together, forming this looped shape. And now, this part of the chromosome and this part of the chromosome, which used to be far apart, are now close together, and so they're more likely to come into physical contact when wiggling around. For the yeast, this is important because changing which parts of the chromosome come in contact actually helps the yeast change its sex. But for us, perhaps this system could be used for engineering applications that require a microscopic material whose shape can be changed to make different parts of the material come in contact at different times.